Can you hear me? Yes. Okay, perfect. Well, thanks very much, David. I think this was a very nice way of setting the stage for the challenges that we have today. My name is Veronique Kiermer. I'm executive editor at PLOS. And I have the distinct pleasure of moderating this conversation here. We have the best part of an hour. I'm going to very quickly introduce the panelists. You've heard from David. Seated next to him, we have next to David, we have Josh Lebar. And I need to refer to all the bios are at the back of the program, so I'm not going to read the bios. But Josh Lebar is at the Biodesign Institute at Arizona State University. Josh has worked in the proteomics field and with antibodies for a very long time and has been a very strong advocate of standards and an advocate of GBSI since the beginning. And he's currently president of HUPO, the Human Proteome Organization. Seated next to Josh, we have Fridtjof Lund-Johansen from the University Hospital of Oslo. And Fridtjof has quite a bit of experience in antibody validation and has designed an antibody array analysis method, which I'm sure he's going to have a chance to talk about. Matthias Hülein has led the Human Proteome Atlas for quite 15 years, and therefore has had a lot. So this is an initiative trying to map the entire human proteome using antibodies, and therefore has quite a bit of experience to share with us in terms of producing and validating antibodies. And finally, Andrew Bradbury, who is at Los Alamos National Lab. And Andrew is an expert in antibody design and recombinant binder design and display strategies. So I think we've got a tremendous amount of expertise on stage, and I would like to start this by having each of them, I mean, David, I think, has told us his piece, but the other four panelists will spend about 5 minutes, and I've asked them to sort of share their own experience of validation, as well as trying to highlight a few points that they think are critical for the next steps and what we're going to do today. And then following that, I'll have a few questions for them, and we'll then open it up to the audience. So let's go in order. I think, Josh, if you want to...
Okay.
Yes,
hello.
Sorry.
All right. So...
You've one slide
yeah, I think
each,
we were, yeah, but
yep.
Dave, I think Mike goes back there. You may want to pull up each of us gets a slide, right? So you're all over
if
it.
We
okay.
Could,
so yes, so I'm not a producer of antibodies, and I'm really just a user. So this is the perspective that I have, and I want to pick up largely on what David McAdams was talking about last night, because I think that is I think a critical element to what we're doing. I don't know if you have my slide or not there. I think that the terminology there are a couple of different terminologies we can use. A lot of what we've been talking about is validation. My background and my research is in biomarkers, and so validation has a very specific term there. You validate a particular test that is predictive of a particular outcome. I would say that what we're really looking for here, or what I would like to see happen here, is developing guidelines for rating antibodies, because I see antibodies as tools, at least in the research space, kind of leaving aside the therapeutic aspect, but focusing on the research space, these are tools. This is just like, if you go into a hardware store, I love doing that. I love buying, power tools. And I'm going to go buy a power drill, right? There's characteristics, just as David pointed out, of what makes that good for me. How long is the battery life? What kind of torque does it have? Do the bits fit well in the kinds of, fasteners that I'm going to use them on? All those different characteristics I want to have rated, right? If it's a tool that is very common, then I can expect a lot from that tool, because there's a lot of different available tools for that. If it's a relatively rare tool, there may only be 2 available, and I'll have to live with what the better of the 2 is possible. So I think that we should be thinking about antibodies the same way. What are the characteristics that they should have, and how should they be rated? And so this is what I put up, and I want to kind of pick up on 2 things that David pointed out yesterday. First of all, he pointed out in the consumer reports that there's like an overall score that they come to, and then he pointed out that they then rate various characteristics. And I think both of those are critical. I think it's important that we think about the various characteristics that antibodies have and rate those. You know, are they sensitive? Do they specifically bind what they're supposed to? Can you detect them in the assay they're intended to be detected in? Those sorts of things. But I think the overall score is really valuable as well. I think there are two things that the overall score does for us. First, it allows users to quickly glance at a list of antibodies and get a sense. I know it's not precise, I know that there may be differences depending on their application, but get a sense of which antibodies are better than others at that particular application. And that way they can hone in on the ones that they want to use. And the other value of the score is it allows us as a community to put value on certain characteristics. We can say that we're going to give way more points for specificity than we might for some other characteristic, because we think that in this context specificity is really important. And so I put together, I know this is long, and we'll just make this available on the website, a couple of key elements. So I think what I would like to see as an outcome of this meeting is that we as a group define the characteristics for each of these application-specific rating scores. So what do we think are the characteristics that should be there for an antibody to do Westerns? What would be those for an IP? What would be those for immunohistochemistry and so on? So that each antibody eventually could be evaluated by that purpose. We need to come up with the scoring system for that. We have to define what the minimal acceptable outcomes are. So if they can't at least achieve that, then they shouldn't even be considered. They don't get a score at all. We can adjust the points depending on how they have fit for various things. I think one of the key elements here is that whatever rating score we develop, the data have to be publicly viewable. So I think if we're going to develop a scheme for this, we have to be able to post on a website somewhere what the images were or what the data were that got that antibody, that score. And I think that not everybody has to rate their antibodies. I think the point of this is that if you get your antibody rated, it's going to be more valuable. More people will want to use it. It'll sell more. And I think that there's an incentive there to having things rated. And I think the last point I'll make on this is that ratings can change. If an antibody gets a certain score and then someone does even more validation on that antibody, presumably it could get better. And as new tools are developed, we can adjust the scheme for how we rate them. The last point I'll make is that I think it's a mistake for us to not do this because we don't have a way of implementing it yet. Because I think we're going to have to figure out who's going to do the rating and how it's going to get done. But I think it would be a huge advantage if we can at least define what characteristics we believe as a community are critical for an antibody to be good at immunohistochemistry, or what are the characteristics that are useful for IP. If we can at least come out of this process with defined rules for what makes antibodies good, that would do a huge benefit for the field. So that's where I'll stop.
I also have a slide. Yeah, so I work with antibody arrays and mass spectrometry, and my aim is to develop a unified pipeline for the two technologies for antibody-based and MS-based proteomics. So I work with a very large number of antibodies. I also come from the medical profession. I'm not an active doctor anymore like David is, but in medicine you have evidence-based medicine, and in there you have a lot of guidelines. And these guidelines are rated. So if you have strong evidence, you give strong recommendations. And strong evidence is typically meta-analysis, systematic reviews, and randomized controlled trials. And then you have weak evidence. If your evidence is only weak, you give weak recommendations. So when we make guidelines, we need to ask ourselves, how strong is our evidence? We also need to ask, how cost-effective are our recommendations? I mean, David was talking about from a clinical perspective, where you can invest a very large amount of money into these antibodies because they're very important applications that are used for patient treatment. We are more talking about the emergency room, where you have lots of people coming in and you don't prescribe an MRI scan for everybody with a headache, because if you do that, you will ruin the budgets immediately. So I think that we need to ask, should the reviewers of our papers always prescribe knockdown, knockout, and targeted MS for every antibody result we publish? Well, if they do, I think it will be very expensive. So I think that we need to explore some more cost-effective alternatives too for validation of a larger number of antibodies. But we need to get the data so that we can make firm recommendations. And Len asked me if he had pissed anybody off. I will give a good try on that now. So I think that we don't have. I don't think that we don't have that data yet. I think that we do. That what we should do in this meeting is really to ask, what don't we know yet, and what do we need to do to find out what we don't know. So then we can, for example, design studies to find out, for antibodies that work in Western blotting, does a single band predict specificity in other applications or doesn't it? And do multiple bands predict cross-reactivity in other applications or doesn't it? These are questions that we can address with well-designed studies. We don't have numbers today. Everybody has an opinion about it, but we don't have numbers. For antibodies that do not work in Western blotting, very, very much more complex situation. What can we do to test their specificity? Well, I think that there are two major technologies that we really need to explore. One is IPMS, and the other one is protein arrays. As of today, we don't know the predictive value or the cost effectiveness of any of them, but I think that we have to do the studies, because if we don't find the answer to that, it's going to be very, very difficult. So again, I hope that we will discuss not only what we are going to do, but also what do we know and what don't we know, what's the quality of our evidence, and finally, what studies can we do to get the evidence that we need? And once we have strong evidence, we can make strong recommendations.
Thank you, Fritjof. Can we have Matthias' slide?
Okay, I should also have a slide. Okay. Okay, good. So, first of all, thanks for some very nice introductions here. I have been working in a wonderful journey in the Protein Atlas for 15 years now. We have generated 60,000 polyclonal antibodies in that, a lot of rabbits. And we have also produced more than 1,000 monoclonals and actually more than 100 recombinant binders. So, and a lot of my time goes to the validation and thinking about validation and forming priority committees and annotation committees and what antibodies should be released and so on. You know, when you have a sort of an elevated pitch on stage, you can only have one or maybe two take-home messages. My take-home message is very similar to David's, and that is that antibodies are application and context dependent. And I want to illustrate that. I guess it's very small here, but to the left we have the paper from Friedman in Biotechniques, which showed what are the applications that are mostly used by research antibodies. And what I have to point out then, that of these 5 most used applications, they all use denaturated proteins. They denaturate the proteins before they use it. But not only that, they denaturate the proteins in different ways. So basically you have the 5 most used applications. In each case you denaturate your proteins in different ways. And just one example, because it's rather extreme, I'm not sure that you're aware that most people that do pathology, immunohistochemistry, they take the sample, they cross-link the proteins with formalin, and then they heat up the sample to 115 degrees. That's pretty absurd for a protein science. They call this epitope retrieval, or we call it. And obviously what we do is that we destroy epitopes and others come up. So it's not so strange that an antibody that maybe works in a therapeutic application doesn't work in immunohistochemistry. And it might not, and an antibody that works in this extreme immunohistochemistry maybe doesn't work in Western blot, where you have actually took SDS, or detergent, to denaturate your proteins. So this is my take-home message. Don't try to sort of say this is a bad antibody and a good antibody. It has to be validated in the context of an application. And this is what I've learned throughout these 60,000 antibodies. And actually, it's interesting, the article that David showed about the poor reproducibility from companies of antibodies, it's actually not that bad, because most of the companies had annotated their antibodies for Western blot, and we use the immunohistochemistry. So actually what we are showing is that most of the antibodies that does work in Western blot doesn't work in immunohistochemistry. And there are this misconception that you can use Western blot to validate proteins for immunohistochemistry. This is so bad, and we just have to go away from this conference and forget about this. You have to validate through the different applications. And this is not very easy, especially for immunohistochemistry. Both David and I know that we spend a lot of time how to do that. It's not easy to do CRISPR-Cas knockdowns because we're working with human material, and you can't really use recombinant tags and so on. So it's not that easy, but it can be done. I just showed you to the right there are some examples. We had a paper a few years ago in Protein Science where we took and really made epitope-specific antibodies towards different targets. This happens to be to an oncotarget called RBM3. And we had 5 then monospecific antibodies that binds different epitopes on the target. And the interesting thing, if you can see the lower one to the right, this is an epitope that seems to work very nice in Western blots, very nice in immunohistochemistry, and very nice in immunofluorescence, ICC. If you go up one, you have an antibody which is wonderful in Western blot, actually, but it doesn't work in immunohistochemistry. So if you use that antibody in immunohistochemistry, you say there's no protein. But obviously, it's simply because the antibody doesn't work in immunohistochemistry. It's not a bad antibody if you use it for Western blot. Actually, it's a fantastic Western blot antibody. And then you have a third one from that works fantastic in protein arrays. It shows very good binding to the target, but it doesn't work in Western blot, it doesn't work in immunohistochemistry, and it doesn't work in immunofluorescence. So with these sort of examples, I just want to teach you that, talking about antibodies is validate them in different types of applications. So this was the challenge when we started the International Working Group 1 year ago in a meeting in Vancouver. We had 1 year because we wanted to propose validations for this meeting. We needed to put them into a framework of applications, and that is the sort of now the 5 pillars that we're proposing, because at least the 3 first ones are relatively easy to actually use in these different applications and then combine them in order to sort of get the validation in good shape. I just want to end by saying, since I've been working so much with antibodies, I can sort of see the fantastic value as all of you in this room. I think antibodies will be more important in the future because we need to move from the genomics, from the transcriptomics into the protein world, even more than we do today. And I can see no better way to do that using the antibodies that we're all so fond of. So again, the 3 pillars that we need to discuss here, or not the pillars, is target specificity, and that has to be application specific. Cross-reactivity, which is very hard because it's context dependent, and then reproducibility, and I can talk for hours, but I probably can talk a little bit more during this meeting. So thank you very much for my 5 minutes in fame here.
Thanks, Matthias. Could we have Andrew's slide, please?
So I'm going to stand because I can't see my slide from the other side of the room. To use a phrase that economists like to say, on the one hand, I was really happy when I saw this slide because what I'm proposing is that we should be using recombinants with public sequencing. But on the other hand, I realized that this actually conflated 2 different problems. So if you go to the next slide, because this wasn't mine, I took the liberty of adding another one. So I think what we're really interested in here is reproducibility and validation, okay? And so if you go on the left-hand side, we have the validation score going from nothing to best practice validation. And on the right, on the top, we have reproducibility. Then it's clear that as you move from polyclonal to monoclonal to recombinant with public sequencing, with public sequences available, you will be improving the reproducibility. But that doesn't necessarily mean it's a good antibody. I mean, it could be a very reproducibly bad antibody, and that's not really what we're interested in. However, if you can get best practice validation with recombinant antibodies with public sequences, then that is going to be the best antibody that you can get. And the reason is that I will use an antibody, I'll publish a sequence, or the sequence is available, and somebody coming 20 years later can try and reproduce that exact same experiment using essentially the same antibody. There are minor issues here that I don't want to go into, like cell lots, lot variations, things like that. But ultimately, the difference between two batches of recombinant antibody are going to be far less than the differences between two batches of polyclonals. On the other hand, if you start off with a polyclonal, which is extremely well validated, that will be a good antibody. But eventually, that polyclonal will run out. And therefore, reproducibility is never going to be as good as when you're using a recombinant antibody. Now, why public sequencing? This is something that the companies have real problems with, and I perfectly understand that. If they give their sequences out, then clearly different people will be able to jump in and make exactly the same antibody, and they've put in all the work for validation. But it's important to be able to identify the exact molecule that you're using. Many of these companies get bought and sold. Antibodies themselves are bought and sold. As antibodies are bought and sold, the names of the antibodies change, so that it's impossible to actually know whether the antibody that you're using is the same as the antibody that somebody else has been using. And that's why I believe that we need to refer to, if not the sequence itself, which is the ideal case, at least to perhaps an accession number, which refers to a secret sequence, or alternatively perhaps a sequence, as Jim Houston has suggested, a sequence that is perhaps put on embargo for 5 or 10 years and then eventually is released to the public domain. I'd also like to say that commentary that was published in Nature was not just my work and Andreas's work. There were 110 co-signatories to that. I tried to make them co-authors. Nature didn't let me. But many of them actually contributed quite strongly to the work. It was Andreas and I that did the bulk of the work, though. Okay, so that's where I'll stop. Thanks.
Thank you, Andreas. So I think you've all raised very important issues, and I think one of the tension that we're going to have between the next 2 days, that we've had in the field for a very long time, is this tension between the usefulness of a validation strategy and the practicality of it, and the feasibility, the economic factors, the access to technology, etc. So to me, this raises a question. When I think about validation, I actually think about two different situations. One is the kind of validations that can be done at large scale. I'm talking about antibody providers, antibody producers, core facilities, service providers, centralized organizations that get some funding, whatever. These kinds of situations where there is really the specialty of the group is to do that at scale. There is access to technology and access of everything. And the second situation is what the end user does in the lab. The boutique validation that you have to do when you buy an antibody, or that you don't have to do because it's been done before. And my question to each of you is, what should we be talking about today? Are these two, is this one of these two approaches that is more important? Are they actually the same thing? Does that provide a different path, a different strategy to actually separate these two things? Are these complementary? Who would like to take this?
So I think I tried to get that across, that they are application specific. And when you said, can you just buy an antibody and start using it without any validation in your lab? And I saw a lot of heads shaking no out there, which I agree with. That is that there's some, if you're using a reagent, there's some level, of quality that you need to check in your setting, in your own lab. And that should be something we think about, that you can't expect, no matter how rigorously validated an antibody is by the vendor, when it comes to your lab, what happens if during shipment it sat in the back of a truck and got to 120 degrees? And you can identify that problem in your lab, but your lab and your usage, whether it's a commercial lab or not, I think is never still has some obligation to prove that the assay is working, and not just with antibodies, with all the other, every laboratory that always includes controls, and that's why we include controls, to do exactly those things. It's a different issue of how should they, are there high throughput methods, and are some methods good enough and other methods requiring more? That's sort of a different issue than I think the point that I wanted to make from that question is that is the end user ever off the hook for just being able to use an antibody in the same way we use distilled water? No.
So I think that's a useful point to keep in mind, because I think that there is guidance that should be directed to that, and it may be different from the guidance that is directed to the overall validation. I think Matthias wanted to.
Yeah, I am quite optimistic about this. There is more than 300 companies that are providing antibodies now. Some of them are very serious, and some of the serious ones are actually in this room. And when I see how they are, and the problem with reproducibility is very much the polyclonals. And what I see is that the serious company take this very seriously. They take a new lot and they validate them from the start, and that is what we have to enforce, I think. But also to have very good identity in also that using new lots and so on. So I'm quite optimistic about that. When it comes to teaching of young students, I think it's very important to teach them that an antibody, a recombinant binder, a monoclonal binder, or a polyclonal, if it's good in one assay, it's not good in the other. Actually, as a matter of fact, monoclonals are actually very, very hard in the sense that they are usually made to the native protein, one epitope, so they are very sensitive to denaturation. So very often you see in literature a good antibody being used in the wrong way. And I think that is what we have to teach the young students and the PIs working with antibodies. But I don't think that we should require them to go through the validation pillars that we are suggesting. These are things that we should ask the providers to do, I think, or some big efforts. Because it's kind of hard and harsh to tell a PhD student that you should first spend 2 years to validate your antibody, and then you can do the experiment.
Yes, and it's not really economically viable. I think Josh wanted to.
Okay, so coming back to your original question, which was whether this is sort of the core facility thing that we're addressing or the end users, and I would say, for my part, it's the end users that I'm most concerned about right now. I think the biggest challenge for most people who run research labs is that there's probably north of 1 million antibodies on the market. When they go looking for antibodies, they have no way of assessing, is this going to work or is this not going to work? We end up buying 4 or 5 of them at a time and having to try them. That's $1,000 minimum to get those just to test them. And I think if there were a way to at least get a first blush assessment that this is likely or not likely to work in the assay that I intended to do, that would make a huge difference here. Obviously, I have to do good science. Obviously, I have to do the right controls. But, and this comes back to, this group, right? Theoretically, we're the experts on this. And so we have Matthias, we have David. They have a pretty good idea of what the guideline should be for an antibody to work well in IHC. They know, if it can do this, and this, it's likely to be good at IHC. And if someone were to set that down in stone, not in stone, if someone should set that down in writing, right? Because it'll change over time. But if we could set it down in writing and say, here are the specific guidelines that today we believe would make a great IHC antibody, that would just do so much for the field. Because now we all have something to measure to. Now we all have a goal to achieve. And we could either ask the producers to do it, we could ask an independent organization to do it, or we could ask just users to do it if they post the data, so we can at least see how they got to that conclusion. So that's where I would come down on this.
I think that this really has a lot to do with what it costs. I mean, if you ask people in this audience who are antibody manufacturers, you can make a list, and see how many steps in this process can you do with your antibody and still make money on it. This is a critical issue. You have to look at what people are actually doing today. They're showing a Western blot, and then they're showing a couple of images of antibody staining. They're not showing extensive validation of, with correlations and all of this. And the question is, can we trust them to do that? Is it cost effective to do it? I don't think so.
So I'd like to go back to the training issue. I think this is really key. So I did my PhD with Cesar Milstein, who invented monoclonal antibodies. I worked on this monoclonal called YD123. It was an IgM, I'll never forget it. I came to Cesar's lab from clinical medicine. I'd been working as a house surgeon and a house physician. And so I was handed this tube. And at the end of every day, I put it back in the freezer, and the next morning I pull it out. And this went on for weeks. And after a few weeks, months probably, I noticed that this tube that originally had been clear was now cloudy. And so I went to Cesar and I said, Cesar, is there something wrong with this? And he looked at me like I was a complete idiot, which to a certain extent, from his point of view, I was. But I just hadn't been trained. I just didn't know. And of course, this antibody had aggregated over years, over the weeks and months that I'd been using it. And because I hadn't been taught to make aliquots, put it in the freezer, and all the rest of it, I think that all students, all PhD students using antibodies should undergo some sort of training course. And this perhaps is an NIH role here. I don't know, maybe a university role. I mean, I think this is absolutely essential because it's unfair to blame users for ignorance if they haven't been given the appropriate teaching in the places where they're supposed to be taught. And in response to Matthias's comment about spending 2 years validating antibody, whilst I agree that's a long time and one would hope to avoid that, it's much better to spend 2 years validating an antibody and get a PhD based on real results than not validating the antibody and ending up with a PhD that is based on crap. So that's my comment.
So going back to I'm trying to have you inform the discussions that are going to go on in the breakout sessions as well today, and really trying. I think we have some disagreements in terms. I mean, Fritjof, you're saying vendors cannot possibly validate antibodies. Matthias, you make the right point that we can't ask every graduate student to spend 2 years, spending and doing that. So I think it's really about these different kinds of guidelines, right? Can we reduce? Did I misinterpret what you said?
I didn't say that. I say that they cannot do that kind of validation. Of course, you could do some kind of validation, and we need to find out what is the most efficient way to spend the money.
Right. So so the point is there a way of reducing the complexity of this validation so that we can have a first approach, a first pass that is cost effective to do at scale, that we can do, and then have different kinds of application specific guidance, and perhaps it's around the kind of control, it's around the kind of, you raise the point of concentration. I mean, all these kinds of, how should an application specific end user validation be done, and what would benefit a first pass evaluation of antibodies at scale? Yes.
Oh, okay.
Josh, yes.
So I think there is a way to do it, and it gets back a little bit to what Fritjof was saying. I think that, so when Len mentioned in his talk that there are really kind of three main things that antibodies have to do, right? They have to bind what they're supposed to bind, they have to not bind anything else, and then you have to be able to detect them in whatever assay it is that you're trying to do. Those are the three main, and then obviously a characteristic of all of those is that they all have to be reproducible and all those other things. So I think if we sat down and we said, okay, we're going to define the guidelines for immunohistochemistry, you could start by saying, okay, what do we need to know via IHC that shows sensitivity, that binds its target? And they could list a couple of different ways, and there's probably more than one way to do it, right? And they range from not too expensive to probably pretty expensive. And you could weight those. You could say, well, if it gets this done by this less expensive way, it's worth this many points to me. This more expensive way, well, it is a little bit better. It's a little bit more accurate. And I'm going to give it a slightly higher score. But I'm not going to go too much higher because, frankly, it's not that much better. Basically, we put our own opinions as to the value of these different things. And you could do the same thing for specificity. You could do the same thing for the ability to detect it in whatever assay it's supposed to be. And I think you could design guidelines that will get you a score for that's relatively inexpensive, that would still show that the antibody does what it's supposed to do. And if you really want to go the fancy route, you get a slightly better score for that, but you definitely invest more to do it.
David?
So I think that there's sort of a tension there in terms of who actually does that experiment and who sets up those first sets of experiments. Because if you are a vendor, you may have a very complex set of first sets of experiments, and that would get a pretty high score. But then a user potentially, depending on who Andrew when he first started his graduate school, not after 2 years in finding the flocculent antibody, but who took some classes but is only in their first half and was told by their PI, here, this is what we do to generate an antibody score, here's the protocol, go at it. I'm going to trust the company more than I'm going to trust that first-year graduate student. And how do you then balance the input from two different experimenters and to give a rating like that? Because that first-year graduate student could then come up with a rating which damns an antibody to misuse, even though it's one of the best ones that company has produced, because they've tested it, but it didn't get published that much, and then it's lost to science, even though it was a really valuable antibody. So I have my own sort of take on this, and is that the person or the body that has the most invested in the production of any antibody is the producer themselves. And so I think that some of us share I think Matthias anyway, shares the opinion that there's some responsibility that comes with that. But the question, and I've heard from, I'm sorry you weren't wearing your name tag last night, but the woman who, from ABCAM, who said they have 85,000 antibodies in their catalog. So obviously they're not going to spend $10,000 a piece on validating, which is, we estimate it costs between $5,000 and $10,000 to rigorously validate an antibody. So somewhere in there needs to be graded and perhaps scored methods for coming up with different approaches.
Fritjof was trying to say
I also
something.
think that we should remember as scientists that there's nothing worse than politicians who make laws that they don't follow. So if we apply very strict guidelines, we must expect that the reviewers will ask us to do exactly what, follow the laws that we make. So we need to ask, how much work do you want to do for each Western blot, each immunohistochemistry image, and each immunofluorescence image? This is what the reviewers, we are now making the laws that we want the reviewers to ask. And we have to remember, what will that cost? How much time will it take?
So I'm the journal editor here, so I get to speak as well. And I think you are very optimistic about the potential of reviewers to be asking for validation. And I do think that that we have, the big lesson we heard last night, and I thought, to me, that was a very important take-out message, is the fact that validation exists and that we agree on what's the best way of validation doesn't mean it has to be mandatory, right? But you take the results with a grain of salt depending on what level of validation it's at. And I think that in a situation as complex as the antibody validation, we need to keep this approach in mind. And as a journal editor, I would be very concerned about reviewers demanding for every single Western blot that an antibody was validated completely. Now, if this is the key to your paper, then there is a reason to do that. And then there is probably a reason to spend $5,000 or $10,000 validating the antibody. But I think that's the approach we need to be careful about. Sorry, Matthias, you wanted to?
No, I agree completely that I don't think we're ready for mandatory rules, and I actually don't think we will in the future either. I see this as a sort of two-stage process. The first process is to lay down what we would like to see for IC or for Western blots and so on, and then require some transparency in papers. So if this is a central antibody for the paper, we should in some way report how we have trusted that this is right. And I think it's more or less being done relatively good. But there is a confusion out there about an antibody working across platforms when they are not. I still think that in the phase 2, I think that we should go and try to do a little bit more vigorous type of pillar validations. What we tried to think about in our committee then was to find things that we could do in a relatively systematic manner. I realize that genetic methods, and I guess most people talk about CRISPR-Cas, it's very expensive. You cannot require the companies to do that for all antibodies. But you could do it there is a lot of CRISPR-Cas type of consortia and so on. And of course, if you combine forces with them, because obviously it's good for the CRISPR-Cas consortia to validate their cell lines with an antibody. So I think still that we could have these kind of public-private partnerships that could be good for the future, but I don't think that we should do it now. Well, we should start now, but we should not require that it should be done. And I think the same thing is true for the orthogonal methods and independent methods that both David and I are very fond of. What you need there is a panel of cell lines or tissues, and I think that could be provided by the companies, so actually researchers can do their own validation without spending 2 years. So I think that there are small ways that the companies can help us to actually do our own validations, and I think that's a very good way. One of the problems with independent and orthogonal methods is that you need variation of your target. And almost half of the proteins in your bodies are housekeeping, and they don't really vary. So actually, these two methods are worthless, because you cannot do a correlation when it's no variation. So this is, of course, a problem, but there is a lot of proteins which has variation, and then you can use them.
I think Andrew went.
So I think there's actually a bit of a confusion here. There's two issues I see. One is characterization. So this is the property of an antibody, what it will do. It will work in Western, it will work in immunofluorescence, it will not work in immunohistochemistry, so on. That is the characterization of an antibody. And the second is the validation. Does the antibody that I've received, does it work now after it's been sitting on the airport in Dallas at 120 degrees? So does it still have binding properties? Everybody's talking about the antibody. And going back to the slides I showed earlier, the problem is that the polyclonal antibody that you buy today is not the antibody or the same antibody as the polyclonal antibody you bought a year ago or 2 years ago. Whereas if it was recombinant, the antibody is the same. Then the characterization data will remain valid for that antibody, and the publication of papers around that antibody will build on the value of that antibody and the data that we know about that antibody. In the case of a polyclonal, ideally, the characterization should be carried out every single time you bleed the rabbit, and that's expensive.
So I think that's an excellent point in terms of the reproducibility as well. And to me, the picture that is emerging is that really we are going to try to agree on best practices to characterize antibodies for specific applications. I think the breakout sessions will be very important. I think we're going to talk about really what are these characteristics, and I think that's also important for these breakout sessions, to really understand what needs to be known about an antibody that you want to use in Western blot, etc.
Can I just make a comment? I'm not sure. I think you are misunderstanding what the companies are doing. They are doing a validation every time they bleed in the rabbit. Every serious company in this room, I think, will do a validation every time they bleed.
That's not what I said.
Excuse me?
We didn't hear the question. Please repeat the question.
Yeah. My question is that we are all talking about validation strategy, but it doesn't really relate to the reproducibility problem. Right, because most suppliers, they do validations and they show the results and still have the problem.
So
I completely agree. We have actually two different questions on this conference. It's the validation of the antibody, which is application specific, and it's the reproducibility. So I agree.
And I think that for the reproducibility, to your point, I want to go to the bottom of that. What are we? So I think the question, if we are going towards exposing more information about these antibodies, we need to be able to identify an antibody and say this is what the tests that were done here are valid for this point. So what information is required for that? Are we talking? We're not talking catalog number. We're talking lot number? We're talking bleed? What is it that we need to be able to go down to? Because I think that from an implementation perspective, and we're straying a bit in implementation here, but that might be very important.
I mean, we will have a reproducibility session later, but I can just say that for me, it's very important to not only have catalog number, but also lot ID. So, and every lot has to be validated as much as the first lot was done. So you really try to repeat. And I think almost all the serious companies are doing this. So I'm not too worried about it, actually.
Just one quick comment. And
but it is a problem because sometimes in literature, you put down the catalog number in best cases, but not lot number.
Exactly.
So when people are reproducing, they don't know which lot they...
And that's what I'm getting at. If we actually want to benefit from the economy of scale of everybody doing validations in their own way, we need to agree on what's the minimal information to identify that this is actually the same thing. And so a quick comment, and I want to spend a few minutes opening this, the floor for questions from the audience. There will be another session following this where we can continue this conversation, so don't worry if you don't get your question in. So really quick comment.
I also believe that every company does some validation when they make a new product. I'm not so sure that they always change the catalog image. And this is the image that we use when we are selecting the antibody.
Right.
Okay. And I'd just like to, again, this issue of characterization and validation is two different things. And I think where they're getting confused here. The characterization is the property of the antibody. Validation is that it actually does that. I think that's important to make that difference.
I'm not confused.
I didn't say you were confused.
I think we have it. I think we have the different stages. All right. A couple of questions from the audience. There is a question at the back there. Please, if you could tell your name and affiliation and keep your question very brief, that would be good.
My name is Mike Browning, and I have a company, Phospho Solutions, that makes antibodies. And in terms of the reproducibility for polys, if you just pool all the serum, then you don't have changes with bleeds, and it's very easy to eliminate that. But I just wanted to say that I really like what Josh was saying about the rating system. I think, and also what Matthias pointed out about if you have lousy Western blot, a lot of times that's a lousy IHC, because I'm concerned with the young people who are trying to buy an antibody. If a company gets a good Western blot, they show it. But if they get a lousy Western blot, they don't usually show it. They usually try to show an image because they know that people who are looking at context-specific validation will be impressed if they see an image. The problem is, image alone at a company website probably means doesn't work in Western blot. So could you talk about that perhaps?
So if I can just make two comments on that. First, I want to address your first point about polyclonal antibodies. And I see polyclonal antibodies as horses. There was a time that everyone moved around the country on horses. And then the car was invented. And now, horses are not gone. There are still horses that are ridden and used, but most people get around by car. And those are monoclonals or recombinants. And I think that we have to, in some ways, if you want to characterize specifically an antibody, if we talk about the antibody and whether we want to validate it here or there, we can't talk about a group of antibodies that may change. As there are for horses, there are uses for polyclonal antibodies today. But similarly, if we're going to talk about specific molecular characterization of an entity, it's hard to characterize a population of entities in that way. And I believe that science is now demanding that level of characterization and quantification that's physically impossible on a polyclonal antibody.
Next question.
Sorry, just quickly, in response to pooling antibodies, pooling polyclonals, isn't that just diluting the good polyclonal antibodies with the bad ones in order to get a consistent response over a long period of time?
Okay, next question.
Yeah, I just wanted to... So Matt Baker, Thermo Fisher. What do you guys think the weight should be for sample preparation in terms of the whole calculation of variability from user to user? Because I think as all of you guys have probably know better than most, that can be a huge factor in the outcome. One lab to another, one hands to another, completely different results.
So I think that's a really good question. I didn't really mention it because we were only one slide, but I think the slide that would have preceded my one slide was that there's a certain amount of information that has to go with before you actually do the rating system, which is what's the SOP that you're following to do the rating. Because I think we can set a set of guidelines, but incumbent upon us as we do that is to set the way that we're going to actually measure those things. And that includes the sample preparation, it includes the lot number, it includes all the different elements of information that need to be made available at the same time that you evaluate and score your antibody.
Richard, you want to do it?
If you want to look at variation in sample preparation alone, look to proteomics and look at the standardization, or rather the fundamental lack of standardization in that field. Yes, so there's a huge variability.
I mean, I would just say, just as a producer, that's one of the things we are challenged with the most. And different scientists that have their own preferences for their own protocols, and using their own techniques and trying to standardize that because it can provide hugely different results.
Transparency in terms of and that is going to be very important as well. I think we also need to be very careful, I think, into what we try to achieve in trying to harmonizing and standardizing compared to what can be achieved just by being extremely transparent about the things that introduce variation. The next question.
Can I just comment on that? So I think it's a very good comment, and it's interesting because what we try to do is to standardize the way we do all the sample prep in different types of applications. But also that means that we're sort of doing it one way and then someone else is using it and doing it another way. So I guess I would hate to mandate how you do sample prep because it would not be possible, I think. But I still think it's just like Fritjof is saying, you have to then report how you do the sample prep because so at least one could reproduce it if you want.
The reality is that most antibodies used these days are provided by producers or companies. And so the companies will have standard operating procedures for sample prep. And I think it's only fair that if you want to use an antibody from a producer, you should use the standard operating procedure provided that producer. That is the only standard operating procedure that they can guarantee will provide the results that they put in their websites.
We have time for one more question.
Hi, I'm Tim Springer from Harvard Medical School, and I'm founding a new institute for protein innovation that will have synthetic antibody libraries displayed on use as a core feature. I want to ask about whether an extreme example of a zoo blot might be useful for characterization. In the therapeutic antibody field, there is definitely a problem with polyreactivity. So, and Genentech is probably the leader in characterizing this. And they have this very simple assay for polyreactivity, which is just to use insect cell lysates, basically an ELISA assay. And the idea is that with any human antigen you're likely to be targeting, it's extremely unlikely that there would be a cross-reacting insect cell protein. And I think that that might be the case for excuse me, that might be the case for all of these antibodies that are to housekeeping proteins. Even those proteins might not, be cross-reactive in a foreign species. I mean, in Genentech they use insect cells. I'm based in Boston, so I propose using another arthropod, the lobster. But you could. You could make tissue sections, from lobsters that you could stain, cell lysates from the appropriate organ. And, it might be just a very cheap, inexpensive way. I mean, like, every company could just do maybe one Western blot and one, example IHC on a this really, really different species. And I think whether this works or not is going to depend on whether the cross-reactivity is really just sort of general polyreactivity and just sort of chance sort of things, or whether there are really specific proteins in there that are, cross-reactive. So I'd like your view on that.
can start. I think generally it's a good idea to have panels of cells and tissues and validate your antibodies through that. So, we haven't really discussed the use of an antibody maybe made for humans, but also see the, the bonding in orthogonal in cross, well, other species. But I support very much that approach. It's very similar to our approach to use panels, put them on arrays, and then try to validate antibodies that way. So I support what it would be a very nice complement to what we are doing, actually.
David?
So I agree that it's got great potential for finding cross-reactivity, but it could still miss things. And the slide that was in my backup deck that I didn't show was 2 antibodies that had been rigorously validated both by the company and in my lab. And then when we ran them on 1,000 patients, there was one antibody that recognized about 10 or 20 patients that weren't recognized by the other antibody. So it was a cross-reactivity that wasn't revealed until we looked at a huge population. So while it works if you find a problem, it doesn't necessarily exclude a problem.
I think we're going to conclude here. Please join me in thanking the panels for all this.